Good day to Dr. Raziv and my fellow classmates. Today we are from Group 2 and we would like to present you the chemical and physiochemical methods to destabilize the cell membrane. First of all, my name is Leong Jun with metric number 202454.
Hi, my name is Nurul Asrafah binti Romzi. My metric number is 2000519.
My name is Lui and Hao with metric number 20023.
My name is Muhammad Hairuzam Samson. My metric is 2000710.
Hi, my name is Sharon San of Bala syndrome, and my matrix number is 202725.
My name is Muhammad Ayub and my matrix number is 202455.
Okay, thanks to my teammates for introducing themselves. Now, I would like to begin our presentation with the introduction first. So first of all, the cell membrane is actually made up of a bilayer of phospholipid, or we call it as phospholipid bilayer, which regulates the in and out of the substance from the cell. And then destabilizing the cell membrane actually it means to lysis or to kill the cell and to extract out the desired content from the cell. And also destabilizing the cell membrane, although it can enhance the recovery of biological products, but it also affects the physical properties of the cell theory and also the downstream processes. In general, animal cells are easier to destabilize as they do not have any cell wall like plant cell. Now I would like to pass my presentation to my teammates for them to introduce the methods of destabilizing the cell membrane. Please.
So right now I will present about the detergents. Detergents monomer have a similar structure like lipid, where it has a hydrophilic head and also hydrophobic tail. In this case, the detergent lipid protein ratio is an important factor for destabilizing the cell membrane. There are 4 stages involved. At stage A, low concentrations of detergents are added to cell membrane. At stage B, the detergent monomer, which are red in color, start to perturb the membrane structurally. At stage C, at concentrations higher or equal to the detergent's critical mixture concentrations, the lipid bilayer break apart, generating lipid protein detergent mix mixture. It is found that to solubilize the integral membrane proteins, a detergent protein ratio of 1:2 is sufficient. At last stage, a further increase of detergent's concentration can cause progressive depletions of the lipid protein detergent mix mixture and cause formations of 4 components as shown in the diagrams. Next, I will pass my turn to Mr. Ravi.
Okay, thank you, Ms. Lui. I will talk about enzymatic lysis. Basically, enzymatic lysis is a lysis process that utilizes enzymes to break down the cell membranes. The enzymes that digest the peptide glycan of cell membranes are collectively called Murian hydrolase. They are categorized as glycosidases, endopeptidase, and amidase based on their bond specificity. Gram-positive cells can be directly exposed to enzyme for lysis process, but for gram-negative cells, they need to be pre-treated with detergent first before exposing to enzymes. The enzyme lysis process is generally conducted at neutral pH and at 35 degrees Celsius. As you can see below is the general process of the enzymatic lysis method. I will pass the presentation to Mr. Hairul.
Thank you, Mr. Raimi. So I would like to go to the next method, which is under solvent. So basically solvent, by penetrating the cell walls and membrane, organic solvent like alcohol, ether, chloroform, benzichloride, and EDTA can disrupt the cells. So basically this is method we known as a chemical lysis. By altering the pH, lysis buffer structure the cell membrane. The aim is to stabilize the membrane protein and rupture the cell membrane to release its content. So this content is such as the DNA, organelles, and many more for the further experiments. So chemical lysis can be classified as alkaline lysis and detergent lysis. That's all from me. I would like to pass to Mr. Ravi. Thank you.
Okay, well, thanks, Hairul. Okay, now I'll present about the last method, which is osmotic shock. So the definition of osmotic shock is a strategy for weakening cells through lysis, which is induced by increasing internal pressure when water rapidly enters cells as the cells are treated to high osmotic pressure. Okay, well, this is caused by a sudden change in the soluble concentration around a cell, which causes a rapid change in the movement of water across its cell membrane. This is also inhibits the transport of substrates and cofactor into the cell, thus shocking the cell. Next slide. So as you can see, there are 2 common conditions for osmotic shock, which the first one is hypertonic condition. This condition is basically water is drawn out of the cells through osmosis and causes the cell to shrink due to high concentration of solute. Next is hypertonic condition, which water enters the cell in large amounts, causing it to swell and burst. Well, those conditions causes the cell wall to break, which is called the disruption of cell. So that's all from me. So now I'll pass to Aksira.
So thank you, Ravi. So let's conclude our assignment. So chemical methods are risky to use for the disruption sensitive cell, as the use of solvent and detergent can cause protein denaturation, damaging the final product. A significant issue related to chemical method is the removal and recovery of the chemical disruptor, making chemical method highly applicable at a laboratory scale. Chemical methods also have low efficacy, making them more expensive and less useful as disruption method. The high consumption of solvent and water makes chemical methods environmentally unfriendly. On the other hand, enzymatic methods are gentle and with fewer side effects, yet high costs make them impractical. So next slide. So these are the list of reference we use to finish our assignment. So I think that's all from us. Thank you.